Hi everyone. Next topic is Human Genome Project.
Before going to the topic, please subscribe my channel, press the bell icon for notifications, like, share among your friends and comment.
The Human Genome Project was an international scientific research project that aimed to determine the complete sequence of nucleotide base pairs that make up human DNA and all the genes it contains. It remains the world's largest collaborative biological project. The idea was picked up in 1984 by the US government. When the planning started, the project was formally launched in 1990 and was declared complete in 2003. The Human Genome Project originally aimed to map the nucleotides contained in a human haploid reference genome. The finished human genome is thus a mosaic, means not representing any one individual. Next we can see what are the goals of Human Genome Project. To identify and map all the 20,000 to 25,000 genes, that is approximate number, in the human DNA from a physical and functional standpoint. To determine the sequences of the 3 billion chemical base pairs that make up the human DNA. To store these informations in databases, to discover more efficient technologies for data analysis, allow the private sector access to the information and technologies that arise from this project. Also, to sequence the genomes of other organisms that are important in medical research such as mouse, Drosophila, etc. Finally, to address ethical, legal and social issues. Many countries were participated in Human Genome Project like United States, the United Kingdom, Japan, France, Germany, Canada and China. Then a parallel project was conducted outside the government sponsorship by the Celera Corporation or the Celera Genomics, which was formally launched in 1988. It is a $3 billion project. Was formally launched in 1990 by the US Department of Energy and National Institute of Health. Human Genome Project was a 13-year-long publicly funded project initiated in 1990 with the objective of determining the DNA sequence of the entire eukaryotic human genome within 15 years. In Human Genome Project, Robert Sinsheimer proposed the idea of sequencing the human genome in the year 1985. Charles D. Lisi and David Smith proposed the budget for Human Genome Project. Human Genome Project Act was passed with the US Congress under President Reagan in 1988. James Watson headed the NIH Genome Program. Francis Collins succeeded James Watson in 1993 as the overall project head and director of the National Institute of Health and was in power until the completion of Human Genome Project in 2003. Jim Kent, a PhD scholar in the University of California, Santa Cruz, in mid-2000 developed a software JIT assembler that allowed the publicly funded Human Genome Project to assemble and publish the human genome sequence. When we examine the timeline, it started in 1970. Frederick Sanger developed a technique for DNA sequencing known as Sanger's method of DNA sequencing. 1985, Robert Sinsheimer at UCSC proposed the idea of sequencing human genome. In 86, US Department of Energy and National Institute of Health came forward to fund. 1989, UK's Medical Research Council joined the Human Genome Project. From that, in 2003, finished version of Human Genome Sequence was completed. Human Genome Project ended with all the goals achieved. So what are the technical aspects of Human Genome Project? The process of determining the human genome first involves genome mapping or characterizing the chromosomes. This is called a genetic map. The next step is DNA sequencing or determining the order of DNA bases on a chromosome. These are physical maps. The mapping strategies. Genetic markers are invaluable for genome mapping. Here, a genetic map shows the relative location of these specific markers on the chromosomes. An example of marker include restriction fragment length polymorphisms RFLP. RFLPs reflect sequence differences in DNA sites which are cleaved by restriction enzymes. Sequencing strategies. To sequence DNA, it must be first be amplified or increased in quantity. Two types of DNA amplifications are cloning and polymerase chain reactions. Now that the DNA has been amplified, sequencing can begin. Sequencing techniques used in Human Genome Project are shotgun sequencing method and Sanger sequencing method. This shows the shotgun method means isolation of DNA. Here, Sanger sequencing method is shown, low throughput but accurate and can handle up to 1000 base pairs, still standard for small scale laboratory use. This is in continuation of the Sanger's method. Outcomes. There are approximately 22,300 protein coding genes in human beings. The same range is as in other mammals. Mouse 23,000 genes approximately, Drosophila 17,000 genes, C. elegans 22,000 genes. We share many homologous genes called orthologs with both these animals, but many of our protein encoding genes produce more than one protein product. A larger proportion of our genome encode transcription factors is dedicated to control elements like enhancers to which these transcription factors bind. The combinatorial use of these elements provide much greater flexibility of gene expression than is found in Drosophila and C. elegans. Gene density 23 genes per million base pairs on chromosome 19, 5 genes per million base pairs on chromosome 13. See, human genome comprises 2% of exons coding region and 98% of introns, that means non-coding regions. Here is applications of Human Genome Project. The sequencing of human genome whole benefit or many field from molecular medicine to human evolution, helps in identifying disease causing gene, identification of mutation linked to different forms of cancer. The sequence of the DNA is stored in databases available to anyone on the internet. The US National Center for Biotechnology Information, that is NCBI, and sister organization Europe and Japan house the gene sequence in a database known as GenBank, along with sequences of known and hypothetical genes and proteins, will allow for advances in agriculture through genetic modification to yield healthier, more disease-resistant crops, benefited the advancement of forensic science. Dear friends, thank you for watching. Don't forget to leave a like and please subscribe my channel.